Welcome. We are learning about the factors affecting expression of the clone genes in the E. coli. Today we are going to learn about the optimization of the codon or the codon choice and the mRNA structure, how it is going to affect the expression of the foreign gene in the E. coli. In the previous video, we have learned about the promoter strength, transcriptional termination, plasmid copy number, plasmid stability, and the translational initiation sequences. The link for this, it has been given into the description box. Now going towards the today's topic, which is the codon choice or the optimization of the codon and the mRNA structure. Before going towards the codon choice, we must know what is codon use bias. Codon use bias refers to the phenomena where specific codons are used more often than other synonymous codons during the translation of the genes, and extent of which varies within and among the species. And this codon use bias, it is observed into the prokaryotes as well as into the eukaryotes. The factors which are influencing the codon use bias, it is the expression level, JC content, recombinational rate, then the RNA stability, codon position, gene length, and other factors like the environmental stress and the population size. Now, here in the definition, what we have seen that the specific codons are used more often than the another synonymous codons. Now, here when you look at this table, okay, here there is the amino acid phenylalanine, and here codon UUC and the UUU, which is encoding the phenylalanine in the E. coli. Okay, so here the phenylalanine, it has the two codons UUC and the UUU. Okay, so if you have observed here now how oftenly it is going to occur. UUC, it is occurring 113 times, and the UUU, it is occurring for the 39 times. Means here the two codons, they are going to encode the phenylalanine, but here the preference it has been given to the codon, and that codon, it is a UUC. That is more frequently used codon, it is whom? It is the UUC, and the UUU, it is used rarely, so it is going to be a rare codon. Okay, so codon use bias means what? When there are synonymous codons, here UUC and UUU, these are the two, they are the synonymous codons, and from this synonymous codon UUC, it is a more often used, so this it is called as the codon use bias. You can see this codon use bias for the isoleucine, asparagine, tyrosine, proline, alanine, and glycine. Now from this table, what you observe that there are the synonymous codons, and from the synonymous codon, some of the codons, they are frequently used, and some of the codons, they are rarely used. The codons which are frequently used, that codons, it has the high amount of the cognate tRNA in the cytoplasm, and the codons which are rarely used, its cognate tRNA levels, they are the lower one. So here the frequently used codons, they are used when there has to be the high level of the expression, and the rare codons, they are used when the genes, they are going to be expressed at a lower level. That is, the proteins which are required in a lower amount in that genes, the rare codons, they are going to be there because they are not allowed, or they are not required to be expressed at a higher level. Okay, and when there has to be the high level of the protein level in the cytoplasm or in the cell, at that time the expression level, it should be the higher one. So there has to be the use of the codons which are used frequently, that is whose tRNA, it is higher into the cytoplasm. In this way, it is not the case that the synonymous codon, they are all the codons, they are not. So here we can say that when there are synonymous codons, all the synonymous codons, they are used, but the synonymous codons, they are used how? According to what the level of the expression it is going to be there. High level frequently used codons, that is there is the codon use bias, and when there is a low level of the expression, there is a use of the rare codons. Now going towards the next one, what is the significance of the codon use bias? Now there are the two main hypotheses. Codon use bias contributes to efficiency and accuracy of the translation and is therefore maintained by the selection. Okay, and the next one, it is a codon use bias exists because of the non-randomness in the mutational patterns, whereby some codons may be more prone to the mutation than the others and are therefore found at a different frequencies. Now, here, at some of the environmental conditions, what may happen that some of the codons, they are prone to the mutations and some they are not prone. So here the choice it is made, okay, the choice it is made that the codons which are more stable, okay, the sequence of the gene which is more stable in that particular environment, that it is going to be selected. So there is a use of the codon use bias. And just now, as I have said that the frequently used codons, they are used when there has to be the high level of the gene expression. That is when there has to be the high efficiency of the translation. At that time there will be use of the frequently used codons, and also for the accuracy also there is the use of the frequently used codons. Now here this codon use bias, where it is used for the optimization of the codon for the foreign gene expression. Now, need of the codon optimization. Why the codon optimization it is required? Because the codon use bias, it is observed to affect the protein structure. Okay, the codon use bias, it is going to affect the protein structure and function through interfering with translation, kinetics, and the co-translational protein folding. Now, here, what we have learned that there are synonymous codon, and from that synonymous codon, some of they are the frequently used codon, that is there is a codon use bias, and some they are rarely used codons. Now here, what is this rare codons? Okay, here we have to concentrate now on where the rare codons. Rare codons, okay, rare codons were defined as codon used at a frequency less than 1%. Okay, the frequency of the uses it is less than 1%. Now here it is the example. Okay, AGG codon. Okay, AGG codon, which is for the arginine, it is used in the E. coli at a frequency of the 0.2%. Okay, means it is a rare codon in the E. coli. But that AGG codon, okay, in the plants, it is used at a frequency more than 1.5%. Means it is not a rare codon into the plants. Now, when we have to express a gene from a plant into the E. coli, at that time what is going to happen, that a gene it will consisting of the AGG codon for the arginine, and that AGG codon into the plants, okay, AGG codon into the plants, it is a frequently used codon, and when that it is going into the E. coli, then it is a codon which is a rare codon. Are you understanding? Okay, AGG it is a rare into the E. coli, AGG it is a frequently used codon into the plants. So here when AGG codon it is going into the E. coli, it is becoming a rare codon. Now, okay, now the gene from the plant, now it has been cloned into the E. coli, as it is now in that gene sequence. What may be there? There may be the presence of this AGG, and now which is a rare codon into the E. coli. And your gene it is under the promoter, which is a strong promoter, and you want the overexpression of the gene. At that time, what may happen? Okay, at that time, what may happen? There are chances of the errors in the translation. Okay, there are chances into the errors into the translation due to incorporation of the wrong amino acid. How it is possible? Now, when there is an ongoing translation, okay, so what is going to happen at the A site? Who is going to come? The charged tRNA, it is going to come. Now, as AGG, it is a rare codon. What is going to happen in the cytoplasm of the E. coli? The tRNA for this arginine AGG, it is going to be a rare one. That is, its concentration, it is a lower one. So the time required for the charged tRNA to come up to the A site, that is at the translation site, it is a longer one because the codon because the cognate tRNA, okay, the cognate tRNA, it is in a lower level. Okay. Now, during this process, what may happen? A wrong charged tRNA, it may come and it can read the codon. It may happen. Okay. And a wrong amino acid, it is going to be incorporated into the polypeptide. And as the wrong amino acid, it is incorporated into the polypeptide, the structure of the protein, it is going to affect. And also the function of the protein, it is going to affect because there is an incorporation of the wrong amino acid. Okay. The here expression, it is how it is going over expression. That is translation efficiency, it is going to be a higher one. And during that, okay, during that, who is going to there? Coming the rare codon, it is going to coming, and as it is a rare codon, it is not going to match the efficiency of the translation. That is the protein kinetic, it is not going to be matched due to what? Due to whom? Due to this rare codon. So there are the mistakes during the translation process, and that it is going to affect the protein structure and the protein function. Or it may happen that there is a lag. Okay, there is a very much lag to come the charged tRNA up to the translation side, and there is a termination, and there is a in between termination, so that what is going to be there is a truncation of the polypeptide. That is, the polypeptide, it is not synthesized completely. Okay, so due to the truncation of the polypeptide, again it is going to affect the protein structure and the protein function. Due to whom? Due to these rare codons. So here, okay, the protein structure function, protein structure and function, it is interfered due to the translation kinetics. Okay, and this it is going to happen due to what? Due to this rare codon. So here, okay, so here when you are going to clone a gene into the E. coli, at that time we have to know about the codons which you are going to add into the E. coli. Like how in the plants now you know that AGG, it is a frequently used codon, but AGG into the E. coli, it is a rarely used. So when you have to express a gene from the plant into the E. coli, then you have to study the codons, okay, the codons on the genes, so that, okay, so that you are able to make the changes into the gene. Okay, you are able to make the changes into the gene. That is, this AGG, it has to be replaced by the another codon which is going to be, okay, which is going to be the frequently used codon into the E. coli. Okay, I think you have understood this. Now going further, now solutions to the problem of the codon uses. Increase in the amount of the appropriate cognate tRNA. Okay, the problem of the lagging, problem of the missing cooperation, or the problem of the truncation, it is going to happen due to what? Low concentration of the cognate tRNA. This cognate tRNA, it can be increased level of the cognate tRNA into the cell. It can be increased. How? How? By modifying the host. Modify the host. Add a gene of the cognate tRNA into the host So the concentration of the cognate tRNA, it is going to increase, or you may have a plasmid vector, and that plasmid vector, it is consisting of the cognate tRNA gene. Okay, so the level of the cognate tRNA into the cell, it can be increased. Okay, and the problem of the incorporation of the wrong amino acid, truncation of the polypeptide, it can be overcome, or there may be alteration of the rare codons to frequently used ones by the sequence-specific mutagenesis, that is the site-specific mutagenesis, or you may also call it as a gene designing, or you may do directly gene synthesis. Okay, you can in vitro, you can synthesize the gene, you can clone the gene. Okay, according to the host, E. coli host, you can add the codons, and that sequence, it can be added into the E. coli host, and you can get a good expression of the gene. Now here, sequence specific mutagenesis or the redesigning of the gene. There are the two strategies for the codon optimization by this method. One, it is a one amino acid, one codon, and the second one, it is a codon randomization. Now, one amino acid, one codon means here assign the most abundant codon of the host or a set of selected genes to all instances of a given amino acid in a target sequence. Now, you can choose the gene which is highly expressed, or you can have a you should know which is the most abundantly used codon into the E. coli. So here, like how the phenylalanine, it is represented more frequently by the UUC. Okay, so all the phenylalanine, it is going to be represented by the UUC. That is, here the codon which is going to be used is a UUC codon. So for one amino acid phenylalanine, one codon UUC, it has been assigned, and all over the UUC it is going to be used. That it is the one amino acid, one codon. So in this way, you have to learn for all the amino acid which is the most abundantly used codon, and that codon, it has to be assigned and allow the expression of the gene. Second one, it is a codon randomization. It uses the translation tables. Okay, uses the translation tables based on the frequency distribution of the codons in an entire genome or a subset of highly expressed genes to attach weight to each codon. Now here, what is this translation table? It is a codon preference table. Now here it is a codon preference table for the E. coli W3110, and here you can see, okay, here it is a AGG. Here it is a AGA, here is a CGU, and here it is a CGC. Okay, these are the four codons which are going to represent the arginine. Now here in this codon preference table, what you can observe that the frequently used codon for the arginine in the E. coli, it is a CGU, CGC, and rarely used it is a AGG. Okay, and here AGA, it is also used. Now these are the four codons for the arginine. So here there is designing of the sequences. Okay, here there is a designing of the sequences, and in that sequences, you are going to use, all the four codons. One sequence, it may use the combination of the CGU, CGC, AGA, AGG. Okay, or in one sequence, okay, suppose you are going to design the genes, okay, about for one protein you can design the number of genes, about 6 to 7 genes you are going to design, and in that combinations of these codons you are going to use and allow that gene sequence to be cloned into the E. coli host. Okay, and after cloning into the E. coli host, you are going to observe that which is the sequence which is giving the high amount of the expression with the accuracy. Okay, with the accuracy, which is the sequence which is giving the high expression, and that sequence it is going to be used, that it is a randomization. That is, randomly you are going to select, you are going to clone. Okay, and allow the gene to be expressed, and by observing which is the efficient sequence, that sequence you are going to choose, and that it is going to be used in the further experiment. This it is the codon randomization. Now here there is no role of the rare codons into the expression of the genes into the E. coli. Yes, there is a role of the rare codons, and that rare codons, they are used during the protein folding when your requirement it is correct protein folding. At that time, rare codons they are used. Okay, you know that there is a co-translational folding. That is when there is an ongoing of the translation. At that time only there is folding of the proteins or the polypeptides. Okay, that it is a co-translational folding. Now, if the efficiency of the translation it is a very high, at that time what may happen, the protein it is not getting, okay, the protein it is not getting the time to fold correctly. Okay, what is the time to fold correctly? Okay, for the correct folding it is not getting any time. So here there has to be the little pause so that the polypeptide it is allowed to fold properly, and that pause it has been given when you are using the rare codons. So when there is a use of the rare codons, at that time required to reach the cognate charged tRNA at the A site, it is a little bit higher than the frequently used codon. So here, when there is a use of the rare codons, there is a pausing, little bit slow down of the translation efficiency, so that a protein it is getting a time to get fold. Okay, so when you are requiring the correct folding of the protein, at that time there is a use of the rare codons. Okay, so it is not the case that rare codons they are not used. Only there is a use of the frequently used codons. It is accordingly what is your requirement. If you are requiring the high level of the proteins, and if you have proper instrumentation for the proper folding of the proteins after extraction and the purification of the protein, at that time there is a no need. Okay, there is a no need of these rare codons. But if you are in need that a little bit correct folding, or you require the proper folding of the protein during the translation only, at that time you are going to use the rare codons. So when you have to express the gene into the E. coli host, at that time you are going to use the combination of the frequently used codons and the rare codons for the accurate protein folding, or for the correct folding of the proteins, and also for the high level of the expression of the genes. Okay, now the next, it is the mRNA structure, how it is going to affect the expression of the clone genes. Now here, okay, the sequences of the 3' untranslated region, that is the 3' UTR of the mRNA, are important. If this region is complementary to the sequences within the gene, okay, if this sequences 3' UTR, if it is complementary to the sequences within the genes, then what may happen? There may be the hairpin loop structures it may form. Okay, so here it is a 3', okay, 3' UTR. Now it is complementary base pairing with the complementary sequences. So what is going to happen now? It is like how the hindrance Okay, it is like a speed breaker. Okay, so what is going to happen? The ribosome now here it is going to stall. It cannot go further. Who is there? The secondary structure it is there. Okay, secondary structure which is a hairpin loop structure it is present. So this ribosome it cannot pass this hairpin structure. So what is going to happen? Yes, there is a no completion of the translation. Okay, the translation it is going to be terminated in between only. So here there will be the truncated polypeptide, and to avoid this, okay, to avoid this, there should not be any complementary sequences in between the genes. No complementary sequences between the 3' UTR, no in between complementary sequences within the genes. Okay, they should not be present, so that there will not be the formation of such a secondary structures. So when you are designing the gene, at that time you have to consider about the mRNA structure, that there should not be any complementary base pairing within the gene or the 5' UTR. It is going to complementary base pair with the gene sequences. This thing you have to keep in the mind. Okay, I think you may have clear the idea about the optimization or the codon choice. Okay, and also how the mRNA structure is going to affect the expression of the genes in the E. coli. If you like my videos, please subscribe my channel and also share my videos with your friends, so the knowledge it is going to spread. Thank you.